Unit 7. Protein. Contents. Protein functions, protein structure, denaturation. Key questions. 1. What is protein? 2. Distinguish between primary, secondary, tertiary, and quaternary structure protein. 3. What are the functions of protein in the cells? 4. Write now on the biological significance of proteins. 5. What is denaturation of proteins? 1. Protein functions. Protein, the purple blobs in the close-up of an animal cell, are the most complicated molecules now. A cell contains thousands of kinds of proteins, which carry out a variety of functions. In most cases, a protein function depends on its complex three-dimensional structure. Click on each protein to see a demonstration and description of its function. Note how function depends on protein shape and changes in shape. Cyanoproteins include hormonal proteins that have coordinated an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals an animal cell to secrete and use sugar. The hormone receptor is also a protein. Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins. Transport proteins carry molecules from place to place. The example shown here allows certain cellulose molecules to enter the cell. Hemoglobin is the transport proteins that carry oxygen in the blood. Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call a response. Storage proteins stockpile building components and that cell can use to make other proteins. Storage proteins instead provide raw materials used by developing plant. Usually, an animal eats them first. Overburning. The main substances in egg white serve as storage proteins for developing chick embryos. An enzyme is a protein that changes the rate of a chemical reaction without a cell being jammed into different molecules in the process. Enzymes promote and regulate virtually all chemical reactions in cells. The structural proteins have many functions, like tens, posts, and rows. They save cells and ensure cell parts. They make substrate drag along which cell parts can move. They bind cells together, making organized units such as muscle, ligament, and the tendons that bind muscle to bones. The silk of spiders and the hair of mammals are also structural proteins. Gene regulatory protein bind to the DNA particular location and control whether on a certain gene will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings. 2. Protein structure. Most proteins are folded into a complex globular shape. A protein molecule consists of one or more strands of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called by polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure. 2.1 Primary structure. A protein has a unique primary structure, a particular number and sequence of amino acids, making the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. Click on magnifying glass in the lower right corner to see primary structure in more details. This illustration shows one of the amino acids making up the three primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone that makes each of the 20 kinds of amino acid unique. Different amino acids have different properties that affect the folding of proteins. Thus, primary structure ultimately determines the shape of a protein, which determines its functions. Secondary structure of protein. In most protein, parts of the polypeptide chain are core or fold, forming threads and corrugations. The secondary structure, the turns and folds of secondary structure contribute to the protein overall shape. One kind of secondary structure in the alpha helix, where the chain threads. Another is platicid, where the chain folds back on itself, or where two regions of chain lie parallel to one another. Click on the magnifying glass to examine secondary structure in more detail. Secondary structure is result from hydrogen bonding between atoms along polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them positive negative charges, and leaving nearby hydrogen atoms with positive charge. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fall back upon itself. 2.3 Tertiary structure of protein. Several reports on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. Click on the magnifying glass to see the tertiary structure in more details. The irregular folding of tertiary structure is result from interaction among the R group of amino acids. Acidic and basic R groups ionize, and three positively and negatively charged groups made from ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic polar R group may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic nonpolar R group cluster on the inside of the protein away from water. Tertiary structure may be the further supplied by strong covalent bonds between several atoms in certain R groups. 2.4 Quaternary structure of protein. Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Click on the magnifying glass for a more detailed look at quaternary structure. Quaternary structure was supplied by the same source of attraction that supplies tertiary structure, hemoglobin, the red oxygen carrying protein of blood. is an example of a protein with quaternary structure. It consists of two kinds of polypeptide strands. Two of a total of four strands make up each hemoglobin molecule. 3. Denaturation of protein. Denaturation is the loss of the specific three-dimensional conformation of protein molecule. The gene may be temporary and permanent, but the amino acid sequence of the protein remains unaffected. If denaturation occurs, the molecule unfolds and can no longer perform its normal biological function. A number of agents may cause denaturation are as follows: heat or radiation, strong acid and alkali, high concentration of salt, heavy metals, organic solvents, and detergents. Testing your knowledge. Answer the questions. 1. Why does denatured protein no longer function normally? Answer. The function of a protein is a consequence of its specific weight loss when the protein becomes denatured. 2. What parts of polypeptide chain participate in the bonds that hold together secondary structure? And what parts participate tertiary structure? Answer. Secondary structure involves hydrogen bonds between an atom of the polypeptide backbone. Tertiary structure involves bonding between atoms of the R group of the amino acid subunits. 3. If a genetic mutation changes primary structure, how might it destroy the protein functions? Answer. Primary structure, the amino acid sequence, affects the secondary structure, which affects the tertiary structure, which affects the quaternary structure, if any. In short, the amino acid sequence affects the shape of the protein because the function of protein depends on its space. A change in primary structure can destroy a protein function. Choose the best answer or best choice for each question. 1. Arrow E indicating or an protein. A. Contractile. B. Receptor. C. Transport. D. Structural protein. Gene. E. Gene regulatory. C is the best answer. 2. Arrow A indicating an or a protein. A. Transport. B. Enzyme. C. Receptor. D. Signal. E. Structural. E is the best choice. 3. Arrow D indicating a protein. A. Gene regulatory. B. Receptor. C. Transport. D. Storage. E. Defensive. B is the best choice. 4. Which of these does not contain of a structural protein activity protein functions? A. Muscle. B. Tendons. C. Albumins. D. Spider cell. E. Ligaments. C is the best choice. 5. Defensive proteins are manufactured by the system. A. Immune. B. Nervous. C. Digestive. D. Integumentary. E. Cardiovascular. A is the best choice. 6. Proteins are polymers of A. CHO units. B. Nucleotides. C. Amino acids. D. Hydrocarbons. E. Glycerol. C is the best choice. 7. What type bond joins in the monomer is protein's primary structure? A. Ionic. B. Hydrophobic. C. Peptide. D. SS. E. Hydrogen. C is the best choice. 8. Which of the electrons is of the secondary structure of a protein? A, B, C, D, E. And E is the best choice. 9. The secondary structure of a protein results from A. Hydrogen bonds, B. Ionic bonds, C. Hydrophobic interactions, D. Peptide bonds, E. Bonds between sulfur atoms. A is the best choice. 10. The primary structure is not directly dependent on A. Hydrogen bonds, B. Ionic bonds, C. Hydrophobic interaction, D. Peptide bonds, E. Bond between sulfur atoms. D is the best choice. Thank you for your attention.